Today we are going to start another lecture, that is gene regulation. That is the second part. Virtually every cell in your body contains a complete set of genes. Means whole body has different genes, but they are not all turned on every tissue. Each cell in your body expresses only a small subset of genes at any time. Means every time all the genes are not active, but they turn on or off for their proper regulation of the work. During development, different cells express different sets of genes in the precisely regulated fashion. Gene regulation occurs at the level of transcription or production of mRNA. For the regulation, for the proper transcription, for the central dogma, they regulate and they are active and they work very efficiently. A given cell transcribes only a specific set of genes and not others. Means there is a code for the production or for the working of their proper sequencing. They only transcribes their key and lock model, like in enzymes they work as a key and lock model. Genetic information always goes from RNA to DNA to RNA, then to protein. That is called central dogma. Every cell has the same DNA and therefore the same genes are transmitted on or off. For example, for the liver, the gene regulation only occurs when the cell genes related to the liver are active or they may on. Therefore, gene expression must be regulated at that point. Gene expression must be regulated in several different dimensions in times, like there are different stages at different times. They are active, they are or on, or they may get off at that time. The development and function of the CNS requires accurate gene transcription control in response to proper environmental signals. For the proper CNS working, there may be a proper environment for the gene regulation, so that they control then the proper appropriate genes, and so gene regulation or gene control may occur at the transcription level. Epigenetic mechanism includes DNA methylation, histone modification, and other chromatin remodeling events are critically important in mediating precise neural gene regulation. So in epigenetic there is a mechanism of DNA methylation, histone modification. Histone is the protein, and chromatin remodeling, that is the part of the chromosome. These are mediate and gene may regulate. One of the striking features of the neurons in their intense utilization of RNA regulatory pathways to perform their diverse cellular functions. And utilization of RNA regulatory pathways to perform their diverse cellular functions, such as axon guidance and synaptic plasticity, that may be occurred by the regulatory pathways by the gene regulation. Neurons express more small RNAs, microRNAs, and alternatively spliced mRNAs than any other cell type. Genome sequencing studies show that diseases such as autism, intellectual disability, epilepsy, that is Mirki in a sense you can say, and schizophrenia in sense you can say, and neural disorder are associated with the mutations in the RNA regulatory proteins, so that these disabilities or these mutations occur, and so the genome may affect. So regulation of the genes may not be get normal, so CNS diseases may occur. Thus neurons are an ideal model system to uncover novel principles of the RNA regulation. So microRNAs constitute a recently discovered class of non-coding RNAs that play key roles in the regulation of gene expressions. MicroRNAs, these are the non-coding RNAs. These are important for the regulation of gene expression. Levels of gene regulation. Here we take an example in the bacteria. In both bacteria and eukaryotes, gene can be regulated at the number of points along with the pathway of the information flow from genotype to phenotype. So compact DNA level of gene control. Take alteration of structures, so released RNA, so transcription then takes place, pre-mRNA forms, then pre-RNA processing occur, leads to the RNA stability, then translation occurs, then post-transcriptional modification occurs, so modified protein active or formed. So, these are the steps involved in the gene regulation. A cell can change the expression of genes in response to the external signals. If there are external signals, so the response may affect. Most of the specialized cells in the multicellular organisms are capable of altering their patterns of gene expression in response to the extracellular cues. If there are extracellular cues, so the gene expression may affect or gene regulation may affect. For example, if we take an example of the liver cell is exposed to the glucocorticoid hormone, that is the steroid hormone, the production of several specific proteins is dramatically increased. If these level may increase, so the protein rise in the formation of the protein rise may occur. Glucocorticoid is the steroid hormone that is produced by the adrenal glands. Adrenal glands may produce glucocorticoid hormone and known particularly for the anti-inflammatory actions. The adrenal gland is the anti-inflammatory. Inflammation means you can say sausage type. That the adrenal gland is an organ situated on the top of kidney. As we know, this adrenal gland is found at the top of the kidney. Glucocorticoid hormone increase blood glucose level. It may also that is a function of the glucocorticoid, you can say, which provides readily available energy for your body cells. Glucocorticoids are released in the body during periods of starvation or intense exercise. Glucocorticoids signal that it may rise the blood sugar level, so they may increase their production in the case of starvation. So glucocorticoid signals through the liver to increase the production of glucose from the amino acids and the other small molecules. How they function on the liver, it may act on the liver and produce glucose from the amino acids and simple molecules. A set of proteins whose production is induced by the glucocorticoid includes enzymes such as tyrosine aminotransferase, that is the enzyme which helps to convert tyrosine to glucose. So it may also help the tyrosine aminotransferase to act on the tyrosine to convert it into the glucose for the increased production of the glucose or a sugar. When the hormone is no longer present, the production of these proteins drops to its normal level. So a gene expression can be regulated at many steps in the pathways. For example, if differences among the various cell types of the organism depend on the particular genes that the cell express at the level in the gene control expression exercised. There are many steps in the pathways leading to the DNA to protein. DNA first lead towards the RNA, then protein, and all of them can be regulated in principle be regulated. Thus a cell can control the proteins it makes by These are the steps involved into the DNA to RNA, then to the protein. Like these steps are controlling when and how often a given gene is transcribed. When there is a for the transcription, this may activate this site and gene transcription takes place. Controlling how an RNA transcribed is spliced or otherwise processed, then RNA splicing takes place, then mRNA, and then selecting which mRNAs are exported from nucleus to cytosol. These are the steps of the gene expression. Then mRNAs are exported, then selectively degrading of the mRNA molecules, then selecting with the mRNAs are translated by ribosomes may translate the mRNAs, or selectively activating or inactivating proteins after the have been made. So these are the steps. First is the transcription control, then leads toward the RNA transcription, because from the DNA the RNA transcription may take place. Transcription control occurs, then RNA processing control, then leads toward the mRNA to RNA is transported, and localization control may occur and then leads to the degradation. Inactive mRNAs are formed, some RNAs are formed, and translation control may lead to the protein activity starts, and protein is formed. Some is inactive protein and other is active protein. These are the eukaryotic gene expression controls in the various steps, leads towards the DNA to the protein formation. Initiation of the transcription in prokaryotes. Eukaryotes are the multicellular organisms. These are made up of many cells. So how transcription takes place in the eukaryotes, from the DNA to the RNA. That is called transcription. Formation of a DNA to RNA is called transcription. How they initiate, how they start up. Unlike the prokaryotic RNA polymerase that can bind to the DNA template on its own, eukaryote requires the several other proteins called transcription factors. Like in a prokaryotes, there is a prokaryotic RNA polymerase that is act on the for the transcription, but in these animals or in these eukaryotes we require the transcription factors. First they bind through the promoter region and then help recruit the appropriate polymerase, then polymerization may start. The completed assembly of the transcription factors and RNA polymerase bind to the promoter, forming a transcription pre-initiation complex is formed by the action of the RNA polymerase to the bind to the promoter, and initiation may occur. Complex is formed. The most extensive study to core promoter element in eukaryotes is a short DNA sequence called data box. Data box is the smaller DNA sequence found 25 to 30 base pairs upstream from the start site of the transcription. Only about 10 to 15% of the mammalian genes contain data boxes, while the rest contain the other core promoter elements. But the mechanism by which transcription is initiated at promoters with the data box is well characterized. 15 to 10% होते हैं इसके अंदर डाटा बॉक्स के mammalian genes के अंदर. Then how the transcription starts. Data box is contains upstream 25 to 30 base pairs of the genes. The data box as a core promoter element. Data box is the core promoter element. That is the binding of the for the transcription factor. Data box binding protein, which is itself subunit of another factor, transcription factor 2, that is TF2D. After TF2D binds to the data box via the TBP 5 more transcription factors and RNA polymerase combine around the Tata box in a series of stages to form the pre-initiation complex. After first promoter is binded, then the transcription factor 2 and TPP RNA polymerase and transcription factor 5 transcription factors bind, then the initiate the pre-initiation complex is formed. One transcription factor 2H is involved in separating opposing strands of the double-stranded DNA to provide RNA polymerase access to single-stranded DNA. This factor is transcription factor 2H form double-stranded 2 RNA in a single strand. DNA into a single strand that is formed as by the RNA polymerase by the aid of RNA polymerase. However, only a low basal rate of transcription is driven by the pre-initiation complex alone. Other proteins known as activators and repressors. Activators activate the reaction and repressors stops the reaction, hinders the reaction, along with the associated co-activators that aid the help the activator. The co-processor that also help the activators are responsible for the modulating transcription rate that regulate the rate of the transcription. Activator protein increase the transcription rate and repressor protein may depress the or decrease the transcription rate or the time of transcription. In eukaryotes, RNA polymerase requires the promoter sequence in the DNA that we already studied. Promoters are the regions of the DNA that promote the transcription. Core promoters are sequences within the promoter that are essential for the transcription. The most common of the core promoters in the eukaryotes in the DNA sequences are the Tata box, that is 5 to 3 or variant TAA 5 to 3 prime or variant found on 25 to 30 base pairs upstream. The Tata box is a DNA sequence found on the promoter region of the genes. Operator is a segment of DNA to which transcription factor protein binds. At the site where the transcription factor binds called the operator region. In the prokaryotic RNA is a core polymerase is a core enzyme consisting of the 5 subunits. These contain RNA polymerase. Prokaryotic RNA polymerase contain 5 subunits. Here we will discuss about these 5 subunits. Before this, we have studied about the eukaryotic transcription. Now we will discuss about the prokaryotic transcription. At the stage of initiation, the core enzyme is associated with the sigma factor that aids finding the appropriate 35 to 10 base pairs of the downstream of the promoter sequence. Here, these are formed at the 35 to 10 base pairs downstream. But in the above case, in the eukaryotic case, we have studied about the 10 to 25 base pairs at the upstream. Like here we will discuss about 35 to 10 and sigma factors are involved. Vector is involved. Now eukaryotic RNA polymerase can only bind to the promoters in the presence of the various specific transcription factors. Tata box is the binding site for the transcription factor called Tata box binding protein TBP, which itself subunits of another transcription factor called transcription factor D. Now enhancer is a short region of DNA. Enhancer is a short region of DNA upstream or downstream of the gene that can bound with the protein to enhance transcription level of that gene's activator. Activator is a DNA protein that regulates one or more genes by increasing the rate of transcription. Repressor may repress the and regulate the one or more genes by binding the operator, blocking the attachment of the RNA polymerase. So repress the activity of the gene expression to the promoter, thus preventing transcription of the genes. We will discuss on the further slides in the later video.